So greetings. Nice of you to join me. My name is Kylar Wiltz, and I'm doing this presentation for the Ethics of Genetic Engineering course. So, looking at the title of my project, you might be, it's kind of funny, kind of odd. Well, the initial topic that I'm working on is how can I develop super durability in skin by using CRISPR technology. So, as I kind of go through my presentation, kind of trying to spark conversation and idea on the topic of how can we use CRISPR to ultimately develop things such as superpowers. I had a lot of fun doing this, and as I previously said, I hope you enjoy. I hope it raised a couple questions in you. So let's begin. So kind of like after introduction, this is my table of contents. We have an additional introduction. Then there's going to be a brief introduction on CRISPR and the way that it works within its own realm. And then we're going to go into the interesting, compelling part of the presentation, which is how can we develop super durable skin and the steps that we could take to essentially be able to do so. So for introduction, Edward Teller once said, "The science of today is the technology of tomorrow." From the beginning of time, man has always found ways to create new innovations. As science has progressed over the years, we've been extremely fortunate to reap the benefits that it's presented us with, whether they come from modern medicines, fuel sources, and everything in between. Recently, the CRISPR-Cas9 technology has come into the forefront in the spotlight. And the big thing about it is, the ultimate potential that it has, not only in medicines, but in other aspects of human research that we have going on. So with something like this, it's a multifaceted topic. And, I was glad that I was able to take this topic and do something fun with it such as super powers. Continuing on, with CRISPR being so new, the biggest thing is trying to aim the trajectory of the potential. With it essentially beginning in bacteria, CRISPR has, evolved into something far bigger. And with that being said, no one fully knows the potential, its trajectory or potential. One thing that we do know, however, is that everyday science and even humans are, evolving, learning new things, doing new things, and moving forward. So with that being said, who knows where CRISPR is going to fall in the next 30, 40 plus years. So, to kind of give you more background knowledge on what CRISPR is, I have a short video here that I would like, to, watch and essentially get like a larger idea of how the CRISPR-Cas9 technology works.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, Researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy.
You see, so now we kind of, got a slight glimpse of what CRISPR actually is through the video. So let's kind of, move forward and get more textual information on CRISPR. So CRISPR are the clustered regularly interspaced short palindromic repeats. First began to be observed in E. coli bacteria in the 1980s. From that point, we've kind of understood that CRISPR was essentially developed by bacteria to fight off viruses as more like an adaptive immunity response. In 2007, it was found that another bacteria had ultimately developed a bacteriophage resistance through the use of its very own CRISPR mechanisms. So at that point, kind of ring a bell within the minds of scientists. So to them, how can we essentially take that technology out, cultivate it, and hopefully manipulate it to do things that could work inside of humans? So from that point, with it being, so easy to get from the bacteria and so cheap, a lot of new ideas and practices have come from this practice of CRISPR. So looking here, on the right side, you have basically the gene editing glossary. You have Cas, which is the CRISPR-associated gene, Cas9, and, a multitude of other definitions that fall within that CRISPR-based information system. And then to the left, you essentially have the diagram that was shown within the video, which is more of like a observation or a figure that essentially breaks down how CRISPR works within bacteria or within the body whenever it's doing its thing. So another aspect of CRISPR is, with so much potential that it has to do good and even bad, a lot of biological and ethical questions have risen, i.e. the reason for the ethics of genetic engineering course that we're currently taking. For a multitude of reasons, CRISPR has become the new hope for future gene intervention procedures. For one, it is far cheaper than other sources of gene editing. It's extremely versatile and widespread with the things that it has the potential to do, because essentially with CRISPR being very specific, you can go around and target almost anything within our genome and do a far more precise intervention than, as say, a gene drive, hypothetically speaking, or things of that nature. So CRISPR right now, because of all of the possible good things coming from it and all the good things in its favor, is now becoming something that far more emphasis is being placed on. Also, it could be used for cancer research, hepatitis, cystic fibrosis, and hemophilia. And I think the biggest thing with CRISPR that a lot of people look at too is that it's naturally found. Granted, man is going in and doing its own interventional things and taking the CRISPR technology outside of the bacteria, but it is naturally found on Earth. So that's a plus that people kind of look into when discussing the CRISPR situation. With all the good things that come with the CRISPR sighting, there are also plenty of bad things. The biggest thing, and one topic that we discuss frequently in classes, with CRISPR being so new and with such widespread potential, we can't really gauge the complete ramifications of what can come with this type of gene editing. And it's not only just from the aspect of the genetic disorders that we're dealing with, or like the genetic problems that it can create. But looking at it from a holistic standpoint, with it being so cheap and easy to produce, how can we control the CRISPR technology from getting into the wrong hands to create, bio war weapons of mass destruction? Someone develops a disease through CRISPR that kills hundreds, thousands, even millions of people. So with all the positives that we had, it's essential to look into the negative aspects of things too. So here's the fun part, the quest to super skin. As I said at the beginning, the superhero that I'm aiming to create is a senior super skin, i.e. the quest for super skin. So hopefully through the next couple of slides, we're able to, kind of bring upon us this conversation of how can we move forward using CRISPR to hypothetically give ourselves a human superpower. So with my project, I'm aiming to examine both sides of the CRISPR discussion and imagine that a CRISPR experimental application, that essentially works well in my favor of my project. Overall, as I've been saying, my experiment is to create a super skin on a human, or an impenetrable exoskeleton for a human being using gene targeting and CRISPR gene editing technology. For me, I've kind of taken a dual route to this. The first one is by essentially creating a super strong exoskeleton using chitin. Now, chitin is found in insects and crustaceans, and it essentially is the main backbone for their exoskeletons and shells. Additionally, I've also taken more of like a non-exoskeleton approach, and just by making your skin extremely durable by creating an influx of strong collagen and keratin within your skin. Through the research and the mechanisms of CRISPR and some of the background information that I found on the molecules that I'm aiming to target, my overall goal is to make my superhero, but with his skills being able to absorb most physical blows and have extremely durable skin, and hopefully that could be attainable hypothetically within these experiments. So let's work on the journey to senior super skin. So our first idea, as previously stated, is the chitin exoskeleton. So with this being the first idea, it was one that's kind of already well known. The building a shell as being one of the main protective coverings that organisms have within our biosphere. So at the bottom, I have a video, just kind of giving us a little bit of background information on what a chitin-based exoskeleton brings to the table in insects, and hopefully taken from this video, seeing how this could apply to humans.
In most insects, the integument forms a rigid exoskeleton that surrounds the outer surface of the animal. The exoskeleton serves a variety of functions. It gives the insect structure, prevents chemical and mechanical damage, protects against invasion by parasites and infection by microorganisms, inhibits water loss, and serves as the attachment point of muscles for locomotion. It also forms the trachea of the respiratory system, forms a lining for the foregut and hindgut regions of the digestive system, and forms the wings in adult insects. This cutaway view shows that the integument consists of a series of layers. The integument is separated from the hemolymph by the basement membrane, a connective tissue layer comprised of glycoaminoglycans and proteins similar to collagen. The epidermal cells are the living part of the integument. Epidermal cells form a monolayer below the cuticle, and they secrete the overlying structural layers, with the exception of the cement layer, which is a product of the dermal glands. Above the epidermal cells is the procuticle, a layer of protein intermixed with chitin. Chitin is a complex polysaccharide comprised of mainly N-acetylglucosamine subunits mixed with some glucosamine, and linked in chemically resistant beta-1,4 bonds similar to the inert beta-glucose of cellulose. Chitin gives the cuticle strength and stability and aids in water retention. The insoluble chitin chains pack closely together to form microfibers of 15 to 30 chains lying parallel to each other and surrounded by protein. The chitin protein chains are deposited in the endocuticle as layers throughout the intermolt period. Pore canals are minute tubular channels that extend from the epidermal cells through the procuticle and end below the epicuticle.
Well, time to come back. I apologize for saying the word wrong. It's chitin instead of chitin. So my apologies. I hope that the video was extremely informative for the topic that I'm shooting for right now. So let's move on. So an application for the chitin exoskeletons that comes with rotation, there are a couple, precautionary things that we, not really precautionary, but steps that we need to take to reach this ultimate goal. First, I want to CRISPR edit the polysaccharide synthase. Essentially, this synthase is gonna, target the nitroacetylglucosamine, which serves a big part within the formation of chitin within these exoskeletons. And as you'll see as we move forward, this is actually something that could possibly be doable because the steps that glucose go through to create chitin also have plenty of similar steps that glucose goes through to produce glycogen or even cellulose, because these three things are derivatives of each other. So that's one thing to keep your eye on. Additionally, there's also the idea of taking supplements of chitin, or let's say eating bugs to get those extra chitin molecules and enzymes in your body from a natural source. Another thing to look out for is the calcium carbonate supplements that you can take. And second to last, we have to increase ATP production for the active transport of these molecules, or the CRISPR edited specific transport for the chitin and the calcium within our bodies that we hopefully are able to produce through the CRISPR. And then let's downregulate the chitin breakdown so accumulation can take place. So let's move forward and go more in depth on these steps. First, the CRISPR modification. So as noted in the first step, the process would take when taking CRISPR into mind. So now we're going to take CRISPR and modify the substrates and enzymes that catalyze the glycosidic bonds of glucose. So as I previously said, within humans, we have something called glycogen. And glycogen is basically a polysaccharide that we use for energy storage. So what I have in mind is, instead of producing the glycogen, my aim with CRISPR is to modify this process of some of the enzymes and synthases that go into creating glycogen, and essentially transform it so that they'll be able to produce chitin. So looking at this diagram here, we're presented with, on the left we have the glycogen pathway, and on the right we have the chitin pathway. So on here you can see that a lot of things are extremely similar. However, I want you to take a look at two things. On the left bottom you have glycogen synthase, but then on the right bottom you have CHS13. So this is one of the main differences between chitin and glycogen. These two enzymes play the huge role in a separatory type of synthesis process. So those are two. Well, the glycogen synthase is one thing that I've intended to target using CRISPR. So let's go to the next slide to look at the second one. So as previously stated, for humans, the CRISPR modification will turn the glycogen synthase into CHS1 and CHS2. These things essentially stand for chitin synthase. So by changing the glycogen synthase into these two things, we're presented now with a pathway that allows the last step of this. So, this polysaccharide modification or transformation, as you may want to put it, will now go from those preceding steps. Instead of going to glycogen, going to chitin. So additionally, CRISPR would also be needed to modify the phosphofructokinase to GFA1. Now, what GFA1 does, since the glucose produces fructose 6 phosphate in both pathways, by modifying the phosphofructokinase to GFA, this will now remove it from the path of going to glycogen and push it towards the path of going to chitin. So another thing that we need to look at is inducing the activities of, a couple more enzymes that are found in our body, because what it'll do, as soon as we, start pushing the glucose, the polysaccharide synthesis, as soon as we start pushing this through and, change these enzymes, if we, push up the induced activity of these enzymes, it'll be more favorable to create the chitin, because now we are creating the substrates that are needed for this to take place. As previously stated, another thing, that will be good is taking additional supplements. The first, as I previously said, is chitin. By, absorbing extra chitin that is already made, let's just say in beetles, insects, that gives us an extra set of already made natural enzymes that could assist inside the process of our own bodies creating chitin. Additionally, calcium carbonate. Now, as chitin usually forms the main base of these exoskeletons, calcium carbonate kind of solidifies it, if that makes sense. So the chitin would create the bricks of everything, but the calcium carbonate comes in and performs as the concrete. So it's a double backing force. Now, the good thing about calcium carbonate is that you can find it in baking soda and other things that you can naturally consume. So that's a good thing. Also, we need to increase blood flow. By increasing blood flow, it'll allow us to get this, new influx of chitin and of calcium carbonate to the areas that we need. With that being said, the inside of our, bloodstream is closer to the dermis. The epidermis is further on the outside and isn't as vascularly equipped as, let's say, the dermis. So what we need to do to be able to get, the chitin and the calcium carbonate to the outside on the epidermis is increase the blood flow through our skin layers to have a chance of reaching on the outside and accumulating to create the shield covering that we're looking for. So kind of, arguing that side of getting things to the dermis, one suggestion that we also could look at with CRISPR is by genetically modifying the active transport proteins. For this sake, I just made an example of the sodium transporter. And what that'll do is, by modifying this to have a higher affinity for chitin and for calcium carbonate, as soon as, it moves through the bloodstream, it has a higher chance of actually getting into the dermis so that it can go to the epidermis. Because these transporters are going to have such a high affinity that as soon as they pass by, the membranes, it'll cycle them in, and that'll give us a better chance of having accumulation at the top or the highest layer of our skin. A side note, one other thing that we'll have to do, as previously stated, is find a way to limit the amount of chitin digestion or breakdown that we have in our bodies. Within our digestive tract especially, we have a lot of enzymes that allow us to break down, certain carbohydrate bonds. By downgrading the capabilities of these breakdown enzymes, it allows for more accumulation of chitin within our bodies, which will be able to assist us hopefully inside of our exoskeletons, because as it'll still get absorbed, but if it's not fully broken down, it'll get into the bloodstream, and then that's when the transporters will be able to cycle them in and just push them out toward the top layer. So here are the pros of having an exoskeleton. For one, you get an extremely durable exterior. Another thing too is that an exoskeleton is kind of a casing around you, so your moisture will always be kept in and you'll never have to worry about drying out. The best part about it is that you get this extra bodily protection. It's like a big shield that's on you. You see from the previous slide, that's why I put kind of like the breastplate. Kind of serve the same purpose as it does in insects and other animals. You have that hard exterior of protection. Now just imagine like the magnitude of strength that it'll have on a human because you're taking in far more immense amounts of chitin. And the extra benefits of having chitin within your body in general, it helps with weight loss, cholesterol control, and another great perk is that it has a lot of provides kidney support as well. So not only is it hypothetically giving you that exoskeleton, chitin is already proven to have these healthy beneficial effects on your body as well. Now with all good things, you're bound to have some, negatives that come with it or cons in this case. The main thing, as previously said at the beginning, is that the unknown outcomes of gene editing. With CRISPR being very new, while we're trying to create superheroes, there are always still these side roads that go away from our main vision of occurring. And by, looking at these things and not knowing what could happen, you could always be like, let's look at the positives more or less than the negatives. But at the same time, if something goes wrong, you have to deal with those consequences. And with not that much research being done on CRISPR, that's always going to be a big issue. Additionally, you have another detriment that comes with the exoskeleton, with it being too rigid. Now, hypothetically, the idea is to have the exoskeleton on top of your top layer of skin, so it accumulate on the outside as like a shell. But with that, it is now a rough surface on your soft skin. So this can cause injuries, lacerations, and collusions on the outside of your skin if it just sits on top of it. Another thing too, with it being so rigid, if it's on top of your rib cage, it may constrict you and affect your breathing to an extent. Another thing, with it being so rigid and tight, it can cause joint pains and limit your movement, and it also could affect your sweating and cooling mechanisms. One of the biggest things, and this is mainly from a social standpoint, is that you don't want to have to deal with the idea of molting and shedding. Humans, we grow a lot, whether it be through puberty, growth spurts, weight gains, weight losses, etc., etc. We are always going through bodily changes. So with that being said, just imagine if every time we do the bodily change, you have to shed an exoskeleton. So that's the biggest thing that you have to look at when from a society standpoint, and you being viewed and always have to dealing with these, what if I just start shedding in public or molting one day? And last but not least, the exoskeleton might be heavy. Just imagine, like a thick plate that's sitting on our bodies the entire time. It may be protected, but at the same time, it's a lot of weight to carry around. So after going over that idea, my second idea is collagen and keratin infused super skin. So for this, idea, I'm aiming at using CRISPR to create a targeted influx of collagen and keratin. By doing this, I hope to create a layer of skin that is both incredibly increasing durability, repair mechanism, and super skin elasticity. Stretching essentially as a means of harm, like something hits you, but your skin essentially stretches to absorb the blow a lot better. So in application, for the collagen keratin super skin to work, these things would have to occur, hypothetically. CRISPR editing of MMP1, the matrix metalloproteinase. This will enable us to alter the enzyme which breaks down collagen and upregulate it to create a collagen at very high and sustainable levels. This could ultimately not only help rebuild skin when it is damaged, but it adds strength and elasticity to our skin. Also, I would like to use CRISPR to stably upregulate keratinocytes. This will cause them to create more keratin within the skin, and by having these higher levels, it'll allow for keratin to accumulate and to have a higher role within building strength within our layer of skin. Another thing I would like to do is find a way to increase skin circulation in most of the levels of our skin as previously stated, so we could push as much of these selected molecules through all of the skin and have a more widespread effect on all of our layers. And implement a skin routine that keeps the skin fresh and moist. With this one, against the exoskeleton, this is going to be our actual skin here. So with that being said, for the keratin and collagen that we already have, you don't want to dry it out or do too much to it. Step 1. So The very first CRISPR modification that will need to be done is editing of the MMP1 gene that produces the matrix metalloproteinase. The proteinase serves as the main collagen degradation molecule within our bodies. By altering this interaction, the aim is to combat the breakdown process and build up the collagen to even stronger levels. It may also be good to take collagen pills at the same time to, kind of place more inside of your body during this time. Continued. The next edit that we would like to target is the increased production of stronger and more stable keratinocytes. So right now we're going to target the KRT1 gene with our CRISPR. Keratinocytes are the main producers of keratin within our bodies and are found within the epidermis. The goal of CRISPR in this part is to basically target that part within the keratinocytes that essentially are the controllers of the energy intake and the production of keratin within the body by essentially creating an environment where you absorb more energy and produce more keratin. We're upregulating the amount of keratinocytes that are producing keratin, and at the same time when you move forward with that, you have more level of keratin in your body, which could accumulate and add more elasticity and more strength to your skin. As I've been saying even the previous one, a good thing to do is to be able to increase circulation of these materials within the body, all at the same time creating an active transport environment that has a large affinity for keratin and for collagen. As previously stated, a skin routine. As even with normal skin, you want to always keep it well moisturized, because when it's moisturized and not dried out, it has a better chance of serving its purpose. When it's dried out, it's essentially dead skin cells, and it begins to die. And another big part and underrated part of this whole thing is having a healthy diet. So the pros of having increased levels of collagen and keratin. With collagen, you have increased bone and joint health. You have better digestion. It helps with hormone and amino acid balancing, and you get healthy hair. With keratin, it helps boost your metabolism and muscle mass, healthy hair, and provides sturdy and healthy skin. But for the most part, and for the aim of this hypothetical project, we want super strong skin. Here are the cons. With everything that comes from the mass influx, there is a strong possibility of something going wrong, especially when considered that CRISPR is involved. You always have to think about the unknown outcomes due to CRISPR. But then you also, with dealing with collagen and with keratin, you have, some negative drawbacks and disease that may come from this. From excess of collagen, you can develop scleroderma. This can cause the skin to essentially harden, and it also can cause organ damage. With excess keratin, you can suffer from chronic eczema, develop calluses, and keratosis pilaris, which is a bunch of small bumps on your body. So, in conclusion, I'm definitely aware that these possible challenges that come arise from a feat of like trying to create a superhero. As I was going through, all of my sources and whatnot, and trying to find the best way to put this, one big thing that I constantly kept coming across is newer and newer questions. Okay, if I influx with keratin, how is it going to react here? How is it going to react here? So my idea here would be in like a perfect utopianized environment. As there may be, other points that you have to look at and consider with this. My biggest thing is, it was having fun with this project, but then at the same time looking at it as hopefully one day being possible. Yeah, this is looking at it as like, very abstract, very, simple and just straightforward. But I understand that it is a very complex topic. The biggest thing for me is just creating it as somewhat of a starting point, even how meager or how quote unquote undergrad as it may seem. But it was a very fun discussion. It really helped my brain fly. At one point, I had 30 tabs open, trying to essentially find a way to go about creating my ideal superhero for my topic. But that's why science is, very rewarding. It's very fun. I learned a lot of things about the human body that I otherwise probably wouldn't have if I didn't get this project topic. But yeah, CRISPR has a lot of potential. Not only, with one day being able to save lives and develop new mechanisms of dealing with genetic disorders, But just having that type of technology and that type of hope of innovation, and being able to have fun with it in this aspect is just something that one day could come to fruition, and maybe one day you'll be able to create superheroes and super soldiers in that aspect, hopefully not for bad means, that is. But just the idea of being able to handle all these capabilities is just you know a beautiful thing. So thank you very much for for your time, and I hope you enjoyed the conversation.